Last time we talked about, the last two lectures were about how we use sequence alignments to find genetic variation when we've sequenced DNA from a diploid individual. What we're going to talk about today is yet another format that those variant callers emit to represent all the sites in a genome or an exome where the individuals sequenced seemingly have genetic variation. And if you recall, the things that it's considering are the depth of coverage, the alleles that are observed, the quality or the associated error with those alleles, strand bias, all those types of things, and essentially using Bayes' rule or Bayes' theorem to assess whether or not the evidence in those alignments is strong enough to conclude that it's true genetic variation and not some sort of systematic error in the sequencing experiment. And when it makes that conclusion, it's confident enough that it is a polymorphic site, those positions get put into this VCF format. It's a complicated format, yet again. We're going to walk through the details of it. And then we're going to talk a little bit about some basic population genetics, something called Hardy-Weinberg Equilibrium, which is super useful for various scenarios. And then one or two toolkits that we can use to work with VCF files, because if we sequence even just one person in this room, we know that we expect something like 4 million sites to be emitted in that VCF file, because any two humans differ at about 4 million single nucleotide change sites in the genome. So if you're looking for genetic variation in a particular gene or genetic variants that meet some particular inheritance pattern, sifting through those potentially 4 million, or if you sequence everyone in this room, probably something like 60 million sites is actually pretty difficult. So we need tools to help us winnow down and isolate particular variants that are of interest. I don't know what to do about this. Okay. All right. So as a reminder, we're starting with FASTQ files. We align them, we get a BAM file, and then we run tools like GATK or Freebase, and we get this so-called VCF file. And VCF stands for variant calling format. I mentioned at the, I guess it was the 2010 or 2009 maybe, American Society of Human Genetics meeting. A bunch of people got together. Most of the people that were at this meeting were listed as authors here. And out of frustration with the inconsistency in the way different variant callers at the time reported genetic variation, they decided to establish a format, a standard, that all these variant callers could use, such that you could compare the genetic variants reported by one tool or one algorithm to another without having to write new code essentially to do those comparisons. Because at the time, this even in 2008, 2009, 2010 really, and still to some degree today, it's a moving target. We're continually developing new approaches to improve the accuracy with which we can identify genetic variation. So having a standard format to assess that increase in accuracy and also just to do our daily work is really essential. So this is a short paper. I think it's maybe 2 pages long. It basically says, hey, we made a format. Here's what the format looks like. We made a piece of software that works with this format. Use it. Okay. So this is a nice cartoon or drawing of 4 genetic variants, and we'll get into the details in a second, for an example VCF file that's on the documentation page for VCF format. Much like the SAM format, which had a bunch of header lines that describe, say, the genome to which those sequences were aligned and the chromosomes that are in that reference genome, the length of those chromosomes, etc. The VCF or variant calling format has its own header. I'm just going to walk through a couple components of it. Actually, do this. The first is this format. So in this example, the data in this file conform to format version 4.0 of the VCF format. I think we're on maybe 4.3 or 4.4 now. So this, even though that first paper described a format, it has been improved since 2011, primarily to accommodate structural variants and other types of genetic variation that aren't these short single nucleotide variants or insertion deletion variants. And this is some bookkeeping. It's basically saying, well, this file was created on July 7th, 2010. The reference genome is this build 36 of the human genome, NCBI 36. So that's two versions back from where we are now. Most people are working with either build 37 or build 38 of the human genome. And then there's a bunch of lines that describe information that's going to be present in this VCF file. So one of the columns, the 1, 2, 3, 4, 5, 6, 7, 8th column is this info field. And for every line in this file after the header lines is a site in the genome where the individual sequence seem to be variable. So there's 4 such sites here. And then for each of those sites, there's a series of information. And a lot of that is carried in this info field. One of the tags that can be present in this info field is this AA, which is in this case representing the ancestral allele. So this header tells you what this token or this ID AA actually means. It has a description associated with it. There's another info field called H2, which is whether or not that variant is present in HapMap. In this case, HapMap is just another study that people refer to. These format fields are pieces of information that are associated with the genotype of each individual. So remember, each of the rows in this file are going to be sites at which one or more individual that were sequenced are polymorphic. So let's imagine we give it 3 BAM files for a mom, dad, and a kid. GATK is going to look at all those BAM files position by position in the genome and look for sites where at least one of those 3 members of the family were polymorphic. When it decides that there is genetic variation among those 3 individuals, it then has to figure out what the most likely genotype is for mom, dad, and kid, or for everyone in this room, or for 1,000 genomes, or for the state of Utah, right? So these columns at the end of the VCF file there's one column for every BAM file that went into the variant calling. So if there's one BAM file per individual and there's 100 individuals, there's going to be 100 extra columns that go on and on, scrolling off to the side of the screen. And for each of those, there's a genotype, which we're going to talk about more in detail in a second. That's this first set of numbers, and then separated by colons, there's a series of other numbers, which those numbers represent various things that are present in this header file. So in this case, this format tells me I should see a genotype followed by DP. I know this is a genotype because I look up GT here and it says genotype. I don't know what DP is, so I can look up in this format field and it says the read depth. So in this case, this individual, sample 1, had a read depth of 13 at position 1 on chromosome 1. And those were the 13 sequence alignments that went into figuring out the genotype for this sample. In contrast, sample 2 had 29 sequencing reads, so over twice the coverage. And so presumably the genotype associated with this sample is more confident than the genotype associated with this sample because there's more sequencing data there. Okay? All right. There's a number of things going on in this file. As you can tell, there's just lots of information here. We're going to try and break it down bit by bit. But basically, the high-level thing that I wanted to tell you is that there's a section of header lines in the VCF format which really give us the metadata or the annotations that help us interpret what's reported in these data lines. And the data lines, there's one data line for every genetic variant that was observed among the individuals we sequenced. Obviously, we need to know the chromosome on which the genetic variant was observed, the position on that chromosome, We want to know what allele is present in the reference genome. That's this ref column. We also want to know what alternate alleles were observed among those individuals that we sequenced, and so on and so forth. Okay, so we're going to now get even deeper into those details. Okay, so let's just look at a basic example. In this case, how many individuals have we sequenced? I guess you've used IGV by now, hopefully. IGV, this is one track for one BAM file, so this is one sample. Okay. GATK is marching along the genome, is board, superboard, superboard, board, board, and then it hits this site and it's like, okay, I have some work to do. It looks at these sequencing reads and concludes that, all right, the reference genome is an A. That's why we see that there. And there's a mixture of A and G reads for this individual. So the alternate allele observed among this individual LF1396 is G. Okay, so now I think by eye you can see why using Bayes' theorem, GATK would conclude that, yeah, there's more than enough evidence to conclude that this individual is polymorphic. So I'm going to report this position in the VCF file. So if you look at this, we're looking at chromosome 7, hence the chromosome here. And if you scan along this, this number line, you can actually see we're at position 11 million, well no, 117,175,373. And that's why this information is in this site. Okay, now, the format for this, the format field just says GT, which means I'm only reporting a genotype for this individual. That's the only information that this very simple variant caller is reporting. Intentionally simple. The column after the format field is the genotype for the sample that we actually looked at that led us to report this polymorphic site. And the genotype is 0/1. The 0 refers to the reference allele. And it's 0/1 because this individual is diploid. The slash is separating the two predicted alleles for this individual. So it's saying, I predict that this individual is heterozygous for allele 0, which is reference, and allele 1, which is the first alternate allele reported. In this case, there's only one alternate allele reported, but imagine we are sequencing maybe a series of tumor genomes, and maybe there's multiple alleles, multiple alternate alleles observed. So it could say ref A, alternate G and T, so you'd have to be able to keep track of which alternate allele the individual is. Okay? Yeah?
So in the last slide, some of the genotypes, they had a pipe instead of the forward slash. Does that represent like haplotype or something?
It does. If you don't mind, I'm going to table that for a second because we're going to get into that in detail. Okay. So if an individual is diploid, there's three logical genotypes, right? There's the individual is homozygous for the same allele as the reference, so-called homozygous reference. So if that were the case, we would have a genotype that says 0/0. The two alleles are both the 0th allele, which refers to the reference. Okay. There's heterozygous, which we just saw, 0/1. And then there's homozygous for the alternate allele. So in this case, 1/1. Okay. There's also a fourth possible genotype. Imagine that we sequenced everyone in this room, and among us, there's really, really solid evidence that there is this alternate G allele at this site in the genome. But for 39 out of 40 of us, there's really good sequencing data. But for that 40th person, we just didn't get great depth of coverage at this site in the genome. So it can be very confident about the genotype about 39 of us, but GATK might say, well, I actually see 0 sequencing reads for, say, me. And therefore, the genotype for me is probably going to be dot/dot. And the dot here represents, I don't know. I'm punting. I'm not going to make a prediction because I just can't be confident about it. All right? And, if we sequence a genome to, yeah, do you have a question?
Yeah, I was going to say, is that like contingent on the filters that you use?
It is. It is. So you could, for instance, you could set a filter that says, I don't want to look at any variant where any individual has less than 5 sequencing reads. So anyone with 4 or 3 or 2 or 1 or 0 would have unknown genotypes. Typically, when you run tools like GATK, you actually don't mess with those parameters. You just kind of accept their defaults, and it uses the data to make those predictions. But yeah, the biggest source of unknown genotypes is just a lack of sequencing data for some individuals. And when you sequence an individual to 30x coverage, that's an average statistic, remember? So most of the sites in the genome are going to be, we did that coin flipping experiment, are going to have, between 5 and 25 or 35 sequencing reads. But some sites, because there's 3 billion to look at, are going to be at the lower tail of that distribution, and some sites will have no sequencing reads. Okay. And so other reasons that a genotype would be unknown aside from depth is maybe there is depth, but perhaps the base qualities associated with the alleles observed at that site are so poor and so variable that it just has no, maybe there's 4 sequencing reads there, and there's an A, a C, a G, and a T, and each of them are base quality 5. Probably a variant caller should say, I won't even hazard a guess. Yeah?
How would the VCF handle triallelic variants?
So it would, if there was a triallelic site, so if we sequenced everyone in this room, most of us are either A or G, but there's some unique individual that is heterozygous AT. What it would do is say G comma T in the alternate allele, and then the 39 individuals that are heterozygous AG would get 0/1 because G would be the first allele listed, and then that one individual who's A/T would have a genotype that says 0/2 because T is the second alternate allele. Okay? Yeah? Oh man, Sarah. Microduplication. Tell me what that is.
Well, just what if the gene, just a portion of the gene duplicated, and so now they've got essentially kind of 3 copies.
Oh, yeah. Same thing. So if there was an alternate allele that was present in that second duplication, and it's not just increased depth, then it would just report it as an extra alternate allele. If it's the same, if the site part of the genome is duplicated, but there's no unique nucleotide within that duplication, it's just going to look like an increased coverage for that individual. So it wouldn't actually, you'd have to use a specialized variant color or a copy number variant color, which we'll talk about later. Good question. Okay. Right. So now we're moving away from just a one sample VCF file into something much more interesting where we're looking at collections of individuals. So let's say we sequence the genome of a mommy and a kid, and we've gone through all these sites in the genome, and we've come to this site, and it looks like quite clearly that both kid and mom are heterozygous, C and T. So therefore, the first part of this VCF file is going to be obvious. It's just the chromosome, the position, the 2 alleles observed, right? I'm skipping quality and filter for now. I'll come back to those in a little bit. So now, what I want to focus on is the fact that you're going to have 2 different genotypes, 0/1, right? But then there's extra information that a variant caller can choose to report. And one thing that's often reported is this AF piece of information. Anyone have a sense of what AF might stand for? Yeah, allele frequency, right? So it's not, allele frequency is not the ratio of reads supporting the alternate allele to reads supporting the reference allele, which would be a rational thing to report. It's actually how many alternate alleles were observed among all the alleles in this study. So in this case, GATK is making the assumption that there's 2 samples, and the assumption is both those samples are diploid. So it's inferring that there are 4 actual chromosomes being represented among this read sampling that we have for mom and kid. And it observes It predicts, however, rather, that mom and kid are each heterozygous, and therefore there's 2 alternate alleles among those 4 total chromosomes, and so it reports that the allele frequency is 0.5, 2 divided by 4. Now, you could have a separate piece of information, which is the ratio of alternate reads to reference reads for mom and kids separately. That information is actually stored in these genotype columns. Right now, we aren't representing it, but essentially you could have additional columns for mom and kid that represent that piece of information. That becomes really essential, actually, especially when you're looking for somatic mutations, because you really want to know, well, how many reads in this tumor genome support this apparent mutation versus how many reads support the reference allele, and that's a proxy for the cellular prevalence of that mutation, sort of the clonal prevalence of that mutation in the tumor genome. Okay, so let's build this out a little bit more and get into a little more complicated example. So this is an example of a VCF file from a study design that my lab and a lot of other labs here at the U would focus on. That is a trio where we've got dad, mom, and kid. If you've never seen these pedigree diagrams before, the convention is a square represents a male, a circle represents a female, and then this tree diagram means these are the dad and the mom, and that's the son. Okay. So what I'm reporting is 4 different sites. This is just a snippet of a VCF file. There should be like 6 million sites in the full VCF. 4 different sites where the reference allele is reported and the alternate allele. What you see here is a mixture of genotypes with, some of them have a pipe symbol and some of them have a slash symbol. The pipe indicates that genotype is, we can know with confidence whether or not the allele came from one parent or the other. And we're going to walk through some examples of how we might know that information. So here's a zoomed in example of a phased genotype. So these up here are unphased. We know what the 2 alleles are from the read data directly. We're just looking at that site, and we know for mom, she's clearly heterozygous based upon the ratio of, say, read supporting the C allele and read supporting the T allele. But at this, we could potentially report the genotype as phased. And so let's go into, and if we can conclude that it's phased, we would change the slash to a pipe symbol. Okay. So how do we actually go about doing that? So what a phased genotype really means is I know with confidence that this first allele, the C, came from mom, and this from mom's mom, from grandma essentially, and this T allele came from mom's dad. The convention is to put, if it is phased, to put the maternal allele first and the paternal allele second. Some tools violate that convention, but that's the convention. Okay, and I've unfortunately reported this backwards on the slide. So there's a couple of ways of doing this phasing. The first is so-called statistical phasing, and this gets back to something that we talked about a long time ago, which is linkage. If you remember the first genetic map that we talked about, maybe lecture 1 or 3 or 4, developed by Sturtevant and Morgan's lab back in the 20s. This was done for fly. The idea is that if alleles are near one another on the same chromosome that's inherited, they tend to be linked or inherited together. Because if they're really close to one another, the probability of recombination between those 2 sites is low. So more often than not, you see, for instance, whenever we see an A in this case, we typically see a G at this next site, right? But that's not all that useful information. In this case, when we see a G at this site, we also see a G. So let's walk through this. This is representing 4 so-called haplotypes that exist in the human population, say. And we're focusing clearly on a very small part of a given chromosome. And what this is saying is that this haplotype, where there's an A followed by a G followed by a C at these 3 different sites, 80% of chromosomes in the human population look like that. 10% look like this, 7% like this, and 3% look like this. And these other sites, these other haplotypes are less common. So we can exploit that information. That's the notion behind statistical phasing. So if we see an A at this site in an individual, we have a pretty darn good guess of what the allele will be at that site. In fact, until we're proven that there's another option, we can basically predict with certainty that that site will be a G, right? But we're less confident about that site. How confident are we? Well, if we give an A, the probability of seeing a C is 0.8 divided by 0.9, because this A, when there's an A, that represents 90% of the chromosomes, so that's the total search space. And then 80% of the time, when we see A, it's a C there. So we can predict, without even having sequencing data at that site, we can predict that will be a C, whatever 0.8 divided by 0.9% of the time will be right, assuming this is perfect information. So we could phase. So imagine that for an individual, this site was reported in the VCF file, and this site was reported in the VCF file. We can phase the A and the C and the A and the G together. We can say that those 3 alleles are probably on the same haplotype. So that's statistical phasing. We're using, it's not certain. We're using basically the frequency of these haplotypes that we've observed to make strong predictions about the probable phase of these alleles observed in the individuals. Does that make sense? Okay. There's a number of tools to do this. It's way beyond the scope of this course to get into all of them. Projects like the haplotype map, HapMap project, the 1,000 Genomes project, and just the more genomes we sequence, the more information we have to make these predictions with really high accuracy. Now, it's one caveat to this is it's fairly robust to predict the phase of 2 alleles based for common variants. Because if they're common in the population, we get a lot of information. We can measure how frequently we see A and C together at these 2 sites. But statistical phasing, or this sort of inference approach, doesn't work terribly well for super rare alleles. So if there's a new variant that you inherited from your parent at this site, right here where my laser is, statistical phasing is really not going to work because that's never been seen before. Okay. There's several reviews on this topic. This is sort of digging deeper into the math of possible phasing scenarios given the frequency of these different haplotypes. And you can refer to this review if you want to learn more. The better way of phasing variants is something I alluded to in an earlier slide, which is so-called phasing by inheritance. When we can directly know the phase, whether the allele came from mom or dad, because we've sequenced the DNA of mom, dad, and the kid. So in this case, dad is homozygous A, mom is heterozygous AG, and kid is heterozygous AG. So we can't be confident, but by quick first glance, we maybe can't be totally sure where the A came from, but actually we can, because we know that the G had to have come from mom. So therefore, the A had to come from dad, right? So if we were to imagine the VCF entry for kid at this site, we would be able to phase A allele to dad and G to mom. So the genotype should reflect it would have a pipe symbol, and it would reflect those two alleles. The ordering would reflect those two alleles that we can attribute to either the sperm or the egg. Same for this site, but the opposite scenario. Dad is now heterozygous, mom is homozygous, kid is heterozygous in this case, and therefore we know that this C allele came from dad and T allele came from mom. Okay. So if you're working on things like rare disease, one way that an individual can get a rare disease is, if it's recessive, is to get what's called a compound heterozygote, where they inherit one, let's say the G allele is deleterious here. Kid inherits the deleterious allele from mom on this site. That knocks out the maternal copy of the gene. And then on this site, let's say C is deleterious, and we can be confident that that C came from dad. So now both the maternal copy of the gene is knocked out and the paternal. So that would be recessive. Recessive diseases require both copies of the gene to be disrupted. And it's this study design of being able to sequence dad and mom and kid that allows us to be certain that it is a compound heterozygote. In the absence of DNA sequencing data from dad and mom, we wouldn't be able to do that, with the exception of if these two sites were really close to each other, within a read length distance, we could actually figure out whether they were inherited on the same allele or on different alleles. But most of the time, these sites are going to be much farther than 150 base pairs away. Okay, so in this case, if we actually look at the genotypes, mom is heterozygous at this first site, dad's homozygous, and kid is heterozygous. But what we've done is reported this alternate allele first because it came from mom. And then at the other site downstream, it's flipped because the convention, as I mentioned, is to have the alternate allele from dad second. Okay? So this pattern of 1, 0 and 0, 1 tells us that there's different phasing, and that's a potential compound heterozygote example. Okay. I mentioned this. Did you have a question? You're right. That is a shortcoming of this slide. They're actually not phased. The parent genotypes actually aren't phased, and I probably should fix that. In fact, I will do that now. Thank you for catching that, actually. So the point, and it's a very good point, is that we really can't phase mom and dad's genotypes because we don't have grandparent information. Thank you. Okay, debugging on the fly. Right, okay, so I mentioned another example. This is a little difficult for me to see on my screen, but makes it up here. Let's imagine that we have sequencing data from dad, mom, and kid here. Kid is clearly heterozygous at this site because we have a mixture of C and G reads. Mom is pretty clearly homozygous for the G allele, and dad is clearly heterozygous CG. Oops. So we can actually phase these two alleles, this site and this site, directly. And I'm going to walk through how. Now, one other caveat here is that this individual, the kid, this appears to be a de novo, a new mutation that's found just in the kid because there's no evidence for it in mom or dad. So it's either something that's somatic, we typically sequence blood. So one scenario is that this read, or this new allele occurred like immediately after the formation of the hemopoietic lineage in this individual. It's like mutation happens in that first progenitor cell, and then it's projected all the way down to every blood cell. Or it happened in either sperm or egg, and is therefore present as a heterozygote in every cell in that individual. With this data, we can't be too certain about that, except that it's somewhat improbable that the mutation happened at the exact first progenitor cell in the blood lineage. So this 50-50 ratio of reads more likely suggests that it actually occurred in the germline. sperm or egg and was not seen in mom or dad because it was exclusive to their germline. Okay, back to the point though. This is heterozygous in the kid. I think we can be pretty confident it's a de novo mutation. Now the question is, did it happen on sperm or egg? And we can actually see, hold on, let me fix this. We can see that this allele and this allele are on the same read. And that read also has, since it has this other allele, we can track that back to dad because that clearly came from dad. So this tells us that these are basically snippets of the sperm gametes haplotype that this individual inherited, right? So we can track this down to this mutation occurred almost certainly on the sperm lineage, or on the sperm. Okay, so I'm going to talk about a couple of resources that we have that really help us to interpret genetic variation and sort of the scale of sequencing, finding genetic variation in the human genome has just gone through the roof over the last few years. It started with the 1,000 Genomes Project, and more recently, this Exome Aggregation Consortium, also known as EXAC. It did exome sequencing of 60,706 individuals. We've talked about this before, but exome sequencing differs from genome sequencing in the sense that we actually, before we put DNA on the Illumina sequencer, we first take whole genome fragmented DNA and hybridize it to probes, oligonucleotide probes that are designed for essentially every coding exon in the human genome. And essentially then we hybridize that DNA and enrich for just the coding sequence of the genome, which is about 2% of the human genome. And therefore, we focus the billion sequences that we get from an Illumina run on just the business end of the genome, the 2% that encodes proteins, rather than spreading out those billions of sequencing reads across the entire genome. Therefore, we get really, really high sequencing depth at every coding exon. And as we learned a few lectures ago, that high sequencing depth really gives us the power to find heterozygotes in those individuals with high confidence. So that study design allowed this EXAC project to sequence the exomes of 60,706 individuals at insane cost, obviously. But it's a really powerful resource for knowing the vast majority of rare and common genetic variants in the human genome. Hey. So one of the staggering results of this study from looking at 60,706 individuals is that there's one genetic variant in the coding exome for every 8 bases. So we talked about on average that any 2 individuals differ at about like 1 in 700 base pairs or something. But that the density of genetic variation obviously increases as we sequence more and more individuals. So in total, when you take all the genetic variation that was found among those 60,706 individuals, there's variants all over the place. Most of those are going to be extremely rare. And so one of the things that I highlighted is they found 9 million variants in the 2% of the genome that encodes proteins. 50% are like 4.5, and now it's more like 6 million. It's like 60% of those variants now, this is a bit outdated, are found as a single heterozygote among all those people. So there's this really huge excess of extremely rare variation in the human genome. So this, they've got all these entries in the VCF file, but if you actually look at that VCF file, the vast majority of the genotypes for those variants are going to be 0/0 homozygous reference, but there's going to be like one individual among those 60,706 that are heterozygous. So that gets it, yeah.
Sorry, going back to the previous slide.
Previous, yep.
Yeah. If the de novo mutation is linked to the dad's haplotype, would it then be a polymorphism?
Great question. No. Not unless this kid goes on to have his, or I guess it's a son, his own offspring, and that allele that he inherited as a heterozygote is then transmitted to, and there's a 50-50 chance that that new allele in the kid, when he grows up to childbearing age, passes it on to the next generation, and then it becomes a polymorphism. Okay, right. So this is a sort of a fundamental population genetics concept. It's something called the allele frequency spectrum, or sometimes known as the site frequency spectrum. The fundamental notion is that most genetic variants in a population, any species, are rare. And that's because mutations are happening all the time. And by definition, if those mutations, as was just asked, are passed on to the next generation, that's inherently a rare variant, right? So this is the breakdown of the variants that were observed in this EXAC study. So 55% of the variants observed were singletons, that is, found as a heterozygote in 1 over 60,706 individuals. Another 35% were found in between 2 and 10 chromosomes, so something like 2 and 10 individuals. So this is super rare stuff, right? I mean, if you went out and sequenced another 100 individuals, it's very unlikely that you'd actually see those variants again because they're so rare in the population. And so 55% plus 35%, that's like what, 90% of the variants observed in the protein coding exome are found in just a handful of chromosomes out of 121,412 chromosomes, right? So this is really important from the perspective of disease. So if you're studying a rare disease, and this was sort of the motivation behind doing this sequencing, if you're studying a disease that occurs in, it's autosomal dominant disease, which occurs in 1 out of 1,000 individuals. You sequence a proband with that disease, and you sequence their parents. You look for all the heterozygotes in that kid that affect protein coding genes. Because if it's autosomal dominant, if you're not familiar, you're basically looking for vertical transmission from one generation to the next. So say mom's affected, she's heterozygous, and kid's affected. We're looking for sites where both mom and kid are heterozygous, and dad is not heterozygous. There's going to be lots of sites like that in the genome. We winnow it down to sites that are in the protein coding exome, and maybe even look at genes that are already known to cause this disease. But if we take a candidate variant that's observed in that kid as a heterozygote in a gene that makes sense, we can now go back to this EXAC data set and ask, well, how common is it in the EXAC data set? If it's more common than 1 out of 1,000 chromosomes, then it's probably not a great candidate, because one of the things I didn't mention is that the individuals in the EXAC study were ostensibly healthy. So if that allele causes the disease and is fully penetrant, then the frequency of it should be much lower than the prevalence of that disease, right? If the frequency of it in the broader population should be much lower than the prevalence of the disease, it should probably be something like never seen in Nomad, for instance, if it's fully penetrant. So this EXAC data set and its successors are really used as a control for identifying genetic variants that might lead to excluding genetic variants that may or may not lead to rare genetic disorders. So why do we have this huge excess of rare variation? There's a couple different scenarios, right? So we talked about purifying selection and adaptive selection several lectures ago. So purifying selection is a scenario where an allele is deleterious for an individual in a certain environment. So if it's deleterious, it's strongly deleterious, that allele should be winnowed away from the population because of natural selection, right? So that's one possibility. There's this huge excess. One possibility is that there's this huge excess of rare genetic variation because most of the new alleles that are present in the human genome are are deleterious, and natural selection is acting very strongly on them and winnowing them out of the population. It turns out that that's not really what's going on. The real explanation for this is that the human population has exploded in size over the last 2 millennia. So if you look back to 8,000 BC, we're talking like, I can't do log ticks here, but let's say like 3 million individuals worldwide. Fast forward to 2000 BC, we're talking about sort of like, 10, 15 million. And now fast forward to the last millennium, we've gone from under 1 billion to nearly 10 billion individuals. So this is on y-axis is on log scale, so you can just see this explosive population growth. And with every new person born, there's 2 meioses. Every meiosis is a genome replication. Every genome replication is an opportunity for mutation. There's about 70 new mutations per meiosis on average. So for every 35 per meiosis, so about 70 per individual. So you generate a lot of individuals over a millennium, and you get lots and lots of new genetic variants. Most of those germline mutations don't actually make it to the next generation, but there's certainly more than enough that do to lead to this huge excess of really rare stuff, just simply because the human population is growing so fast. And it's kind of the same thing with like Drosophila, for instance. There's just, I have no idea how to even estimate how many fruit flies there are in the world, but there's lots of them. And it's just, constant population growth. So lots of new alleles, which is why there's so much genetic diversity among them. Okay. So what I want to cover for the last little bit of the class is this fundamental notion called Hardy-Weinberg Equilibrium, and it actually is another application of the binomial distribution. Okay. So it can generalize to triallelic and tetraallelic sites, but it's simplest to think through in the context of a biallelic site in a diploid organism. So the naming convention is to represent allele frequencies, the 2 alleles, as P and Q. P, the frequency of one allele, and Q, the frequency of another allele. So in this toy example, we've got 6 chromosomes. 4 of them have an A, 2 of them have a G. So the frequency of A is represented by this P frequency, so that's 4 out of 6. The frequency of G, we could compute in 2 ways. We could actually measure it directly, or we could basically infer it. So if we know the frequency of one allele and there's only 2, it's just 1 minus the frequency of the first allele. So the assumption is that P and Q add up to 1, as they do in this case. So P and Q just represent how many chromosomes have one allele versus how many chromosomes have another allele at a particular site in the genome. Okay, Hardy-Weinberg equilibrium states that essentially, in the absence of selection, in the absence of a bottleneck, in the absence of any selective pressure at all, if we know the frequency of the alleles, P and Q, we can predict the frequency of the 3 possible diploid genotypes given those allele frequencies. Okay? And then the beauty of it is we can then go and measure those genotype frequencies by sequencing people. And the null hypothesis is those genotype frequencies will be in equilibrium. That means those genotype frequencies match exactly what you'd predict based upon these allele frequencies. If they don't, it's evidence of other things potentially happening, potentially purifying selection, potentially inbreeding, potentially strong migration or bottleneck effects. Okay, we don't really know what would lead to that deviation. We'd have to do further research to study that. But deviation from Hardy-Weinberg equilibrium is sort of a crude attempt at looking for things like selection, migration, bottleneck, or it's also a quality control thing. You could have sites in the genome where the genotypes, it's hard to genotype accurately, So you get this deviation between the apparent allele frequency and the observed genotype frequency. So it's also can be used as a nice quality control measure. All right, so let's walk through the math. If we have these two allele frequencies, so the frequency of A and the frequency of G, represented as P and Q respectively, we can use binomial distribution properties to estimate the frequency of the three different genotypes. So the frequency of a homozygote A is just the frequency of the A haplotype squared. So it's basically this gets back to basically Punnett square stuff. So the probability of seeing a homozygote A is the frequency of A times the frequency of A. So we expect 44.89% of individuals to be homozygous AA. We expect, just plug and chug here, 2 times P times Q, 44.22% of individuals to be heterozygous. And we expect a much lower fraction of individuals to be homozygous for the alternate allele. Okay? And these genotype frequencies also have to add up to 1. Okay? So if we sequenced 1,000 individuals and went and looked at this site, we'd expect 448 individuals to be homozygous A, 442 to be heterozygous AG, and 108 to be homozygous GG. Okay? And you basically can, plug this in for any allele frequency. So if the two allele frequencies are the same, half of individuals are P, half are Q, then this is the breakdown. Now when we get to really rare variants, so like this, let's say this G allele, the A allele is super rare. It occurs at a frequency of 1 out of 1,000 chromosomes. Then the probability of seeing a homozygote for that is very, very rare. And this poses a problem. So this is 1 in a million frequency. So we'd have to sequence probably at least 1 million individuals to even ask the question as to whether or not the observed number of genotypes is less than what's actually expected. And we can't do that. So for very rare alleles, we really can't leverage the Hardy-Weinberg equilibrium until we sequence like everyone on Earth, which probably won't happen. So if we sequence, given these frequencies, if we sequence 100 individuals, how many heterozygous AG individuals will we expect? 18, that's right. How many AA homozygotes? 1, right? And 81 GG homozygotes. So there's this nice plot that sort of represents the genotype frequencies that we expect. So this homozygote Q, heterozygotes in red, and homozygotes in blue that we expect as a function of the allele frequency of the first allele, because we can infer the allele frequency of the second allele if we're given the allele frequency of the first allele. Okay. So let's walk through an example. So the frequency of some allele Z at a given locus on chromosome 7 is 0.3. What proportion of individuals do we expect to be heterozygous for Z in this other Q allele? What's the equation we would use? So let's call the Z allele P. What's the frequency of P? 0.3. So what's the frequency of Q? 0.7. So you can do the math. We got 0.3 times 0.7 times 2. What is it? 0.42, right? So I think what's quite nice about this is that it's very simple math to just plug this in. Okay. Now I mentioned I just want to touch upon this again. We can use those allele frequencies to estimate the genotype frequencies, but really the interesting bit is testing whether or not the observed genotype frequencies match what we expect. And if they do, we can say that that site is in Hardy-Weinberg equilibrium. But what if it's not? So-called deviations from Hardy-Weinberg equilibrium. And a clear example would be a recessive disease. So if that variant leads to recessive disease, the homozygotes that we observe for that deleterious allele should be far lower than we expect given the allele frequency, because heterozygotes are tolerated. So what we should see is an inflation of heterozygotes, observed heterozygotes, and a deflation or depletion, I guess is the better word, of homozygotes for that deleterious allele. But the problem is that there are other potential explanations for that depletion. Inbreeding, bottlenecks, positive selection, purifying selection. So maybe the other allele is positively selected, or that allele is purifying selected in the case of a recessive disorder. Or it could just be randomness. There are just sites that are under drift and sort of we can observe deviations from the expected allele frequencies. Question? No. Okay. All right, so let's take another example. Let's say a population is in Hardy-Weinberg equilibrium, and the frequency of a recessive homozygote genotype for a certain trait is 0.16. So that's the genotype frequency. What's the percentage of individuals that are homozygote for the dominant allele? 0.36, yep. Okay, so what I want to move into now in the last few minutes is we've talked about VCF files, we've talked about Hardy-Weinberg equilibrium, and now we're going to just introduce one more tool set for actually working with these formats. So it's another tool out of this group called HTSlib. There's a bunch of developers, really excellent developers that make these tools. And it's called VCF tools. Why is it not called VCF tools? Well, there is another tool called VCF tools, and that's actually quite great. But VCF tools is a more flexible, faster, more general tool set. And the B stands for binary. So this is the same kind of distinction as SAM versus BAM. There's a binary version of VCF format called BCF. And there's a bunch of different subcommands for this, as you've learned. There's subcommands to filter variants, to winnow down to variants that meet certain inheritance criteria, or on particular chromosomes, or in a particular chromosome range. Like maybe you know that the gene that you're interested in is from position 1 million to position 1,100,000 on some chromosome. You can use this filter or other commands to winnow that down to those sites. So another tool that comes out of the BCF tools package is this query command, which allows you can, in this command, for example, will take this file that's in BCF format. It's very easy to convert a VCF file to BCF format. And it'll just report the position column. So that's that second column that we saw in the B. So, this is kind of a boring command, but it will just tell you the position of every variant on every chromosome. You could, and there's really great documentation up here. I would encourage you to look at that documentation. It'll be useful for the homework. But it allows you to generate commands that allow you to cut out the information from this complex VCF format and put it into a file that you could load into R or put in a spreadsheet and manage more directly. As I mentioned, you can filter variants based upon different criteria. So in this second command, it's saying, all right, I want you to print out the chromosome, the position, the quality, and the depth from this VCF file for every variant where the quality score is greater than 20, and that's what these two ampersands mean, the depth, the total depth observed across all the individuals is at least 10. So this command is really helping you to identify high quality variants based upon this criteria, okay? And just get out the position and the quality associated with them. You could extend this command -f that stands for fields. What fields from the VCF file did we want to extract? So you could add %ref, and that would also print out the reference allele. %alt, that would print out the alternate allele as well. And this hopefully gives you an example of how to spit out a text file that you could then use with tools that we're more comfortable with, like grep and other things. Okay. So the homework is at this link. Obviously, it'll be due on February 20th. And the main goal of this homework is to get you to start working with VCF files and really getting your hands dirty with the data and trying to identify variants of potential interest. Yeah?
You know, that's only 2 days after homework 4 is due.
Oops, I meant to fix that. Thank you.
Homework 4 is due next Tuesday, but going to class on Monday, so we don't have any kind of CTAs before, unless we go tomorrow. So could we move it to the 20th for homework 4? Or?
We've had like 3 weeks, right? Yeah, I think let's keep it where it is. I would use the TA time now. Okay, so this one will be due on the 27th. I'll update this link, and next time we'll talk a little bit more about the VCF format and other aspects of it. Okay, thank you.